Next speaker is Dr. Laskai, and he's going to talk about cell physiological effects of reduced deuterium content in the leaves of Alodia canadensis. I hope I said it right.
Good afternoon, ladies and gentlemen. I have come from Szeged, and perhaps for those who are not familiar with the geography of Hungary, it's worth having a look at the map. Here we are now in Budapest, and if you travel about 170 kilometers to the south or southeast, you can come to Szeged. But if you travel a little bit more, about 200 kilometers, you will get to the border. So Hungary is a relatively small country, and 200 kilometers can be considered as large distances. It's a relatively nice university town. Here in this place we have an open-air theater all summer, and this is the building where the high-rank officials and the rector of our university have their offices. And the population is about 160,000-70,000 people, but we have 30,000 students, and so you can appreciate how lively the city or the town can be, especially in term time. We often say that the whole town is a university campus. So anyway, we all know that plants are beautiful creatures because they produce food for us, either directly or indirectly, and also they produce oxygen for us, without which we couldn't liberate the energy which is stored in the food molecules which we consume. But of course, but in addition, plants provide us with interesting experimental system with which we can study basic cell physiology, and for example we can study the effect of deuterium depletion. Especially good or promising experimental objects are the aquatic plants, which are in close connection with the surrounding water, and they do both photosynthesis and respiration with the constant contact of their ambient water. So if they photosynthesize, they take up CO2 from the water. If they respire, they release CO2 into the water. So they might be useful objects and experimental systems for the studies of deuterium depletion. Perhaps not all of you are familiar with what Alodia is, but thanks to the morning session, we know that Alodia is a favorite aquatic plant, and is a submerged microphyte which now is present also in Europe. It originates from North America, and there were times when it was not a very nice guest arriving from the United States. In the 50s and 60s, it caused considerable problems in the waterways, many waterways of Europe, because when it brought into Europe, into Germany, it coiled around the screws of the boats on the River Rhine at the good old days when there was some wildlife in the River Rhine, and it caused stoppages and tie-ups. And in Hungarian, its name is átok-hínár, which means cursed water weed, and perhaps some of these problems can be associated with this earlier behavior. But now we can find this plant in pet shops, and all those people who have aquaria at home are familiar with it. Apart from this plant is very easy to work with, because the leaves can be stripped off very easily and can be put into any kind of experimental media, and then the physiological actions can be easily studied. this is actually this plant, which is a useful object for cell physiological studies as well. Have lots of chloroplasts in the cells, and all kinds of cellular studies can be carried out with it. So, as you know well, the plants can carry out photosynthesis and respiration. In the light, they carry out at the same time, both of them. But photosynthesis usually predominates, and in the light, plants are capable of fixing CO2 and producing sugar, and they are capable of utilizing the energy of photons. In the dark, they only respire like us, and with respiration they produce energy, ATP, so the plants are the only creatures which can carry out both different kinds of energy supplying cellular mechanisms. So the chemists were able to measure how CO2 can dissolve in water, and they were able also to measure the rates of the different reactions which are taken place when CO2 gets into water. And according to these measurements, there are different components which are formed when CO2 is dissolved in water, including carbonic acid, which can dissociate into bicarbonate, which can also dissociate into carbonate. But according to the speed of these reactions, it can be considered that at or near neutral pH values, this reaction is sort of fast. This reaction is very slow. This predominates only at high pHs. And so, therefore, the rate-limiting step is this value, this reaction. Therefore, for the sake of simplicity, at around near pH values, we can consider the dissolution of carbon dioxide in water according to this equation. And the three important components, the presence and the ratio of the components is a function of the pH, and you can see the fraction of CO2, carbonate and bicarbonate and carbonate in the function of pH, and this is the pH around 6.5, which we chose for our studies because that's the pH when half of the carbonic acid or half of the carbon in the water is present in CO2 and half of it is present as bicarbonate, so that the plants can take up carbonate without any problem. And if we put the plants in the water and switch on the light, they will start to photosynthesize and they will take out the carbon dioxide, and this will change the equilibrium, of course, and according to the basic rules of equilibrium kinetics, the reaction rate of the backward process will increase, and this will cause a decrease in the proton concentration. In other words, in the light, if the plants start to photosynthesize, we can expect a decrease in the pH, decrease in proton concentration, so an increase in the pH, and the reverse reaction is expected in the dark.